The overall goal of this protocol is to execute in vitro and in vivo deacetylation assays in order to establish the role of proteins as specific deacetylation substrates for Sirtuins.
Despite the increasing number of proteins found to be acetylated, arguing for a widespread presence of acetylation, specific enzymes regulating this modification have not yet been identified for most of these proteins. The main advantage of these assays is that by establishing a protein as a legitimate substrate of a specific deacetylase, novel roles and functions of the deacetylase can be unraveled.
So, this method can provide insight into the identification of Sirtuin-2 specific substrate. It can also be applied with minor modifications to study focus on other members of the Sirtuin family.
To begin this procedure, prepare a 10 cm culture dish of HEK293 T cells in 8 ml of DMEM with 10% FBS and antibiotics, so that the cells will be about 70% confluent after growing overnight. On the following day, co-transfect the cells with the following: 4 micrograms of lentivector expressing GFP and flag-tagged SIRT2, 4 micrograms of packaging vector, and 0.5 micrograms of envelope vector. Use polyethylenamine, or PEI, at a ratio of 3 microliters of PEI per microgram of DNA. 12 hours after transfection, change the medium.